A pleasant day to each and every one. For today, I am going to present the results of the research activity I have conducted for the course Laboratory Techniques for Biological Studies. And this study is entitled Phytochemical Screening and Analysis of the Active Constituents on the Leaves of Nephelium lappaceum or Rambutan's leaves in comparison to the commercialized tea using Thin Layer Chromatography Technique towards the identification of the plant's potential as an herbal tea. To give you a glimpse of the research endeavor for this research activity, in today's time where alternative medicine is rampant and observable to traditions and cultures of many people through the practice of boiling leaves and consuming it, believing that it is medicinal and beneficial to human health. This study have investigated the potentiality of Nephelium lappaceum lives as an herbal tea using phytochemical screening and analysis through thin layer chromatography technique and by comparing its present active constituents to the commercialized tea. The commercialized tea that has been utilized as a point of comparison for this research activity is the Lipton Yellow Label tea. This study has employed a quasi-experimental research design and seeking to validate and explore the presence of active constituents in the leaves of Nephelium lappaceum in comparison to the active constituents on the commercialized herbal tea. At the end of the study, it has been statistically proven that there is no significant difference between the test substances at 0.05 level of significance based on their RF values or the relative flow values and the presence of specific compounds and its implied biological significance to human health. In the study of Morilla et al., as cited by Agapine on 2019 for herbal medicine, the leaves are the most widely utilized component of the plant. One explanation for this, unlike stems and roots, leaves are the easiest to pluck and preserve the completeness of the plants because they are easily regenerated. Furthermore, vital chemical substances like tannins, essential oils, and flavonoids are preserved in high concentrations in the leaves. This might be the major reason why people around the globe of different traditions and culture are practicing decoction as extraction in order for them to consume and get the beneficial or health benefits of drinking plant extracts. Claverie et al., on the other hand, cited that tea production is not yet prevalent in the Philippines due to known reasons like climate, soil acidity, and other issues. Herbal infusions, on the other hand, are a type of tea popular in the country. And of course, like what I have said a while back, decoction in the Philippines is also popular, just like boiling the leaves of guava, avocados, and the like. So in order to test a plant's potential as an herbal tea, it should contain the active constituents that are highly beneficial to human health. As Chandrasekhara and Shahidi 2018 pointed that natural bioactive components found in herbal teas and beverages include the following: carotenoids, phenolic acids, flavonoids, coumarins, alkaloids, polyacetylens, saponins, terpenoids, among others. Natural bioactive compounds have a variety of biological effects, which includes the following. And it is going to be according to a large body of scientific evidence. In supplementation, Bag et al. On 2022 stated that tea is made out from the leaves of Camellia sinensis plant, which is one of the most extensively consumed drinks. So tea's promised health benefits have been related to its several phenolic components. And one of these phenolic components particularly is the polyphenolic compounds or components. And tea also includes antioxidant flavonoids, alkaloids, polyphenols, theanine, and other compounds that have been linked to a range of health advantages. Peluso and Serafini on 2017 further reiterated that this high level of bioactive compounds like polyphenols is assumed to be responsible for the health advantages attributed to its use and consumption. As such, it can also be inferred from this that polyphenols can be a ground of basis if a plant is going to be considered or has the potential to become a herbal tea. In the study of Abhishek et al. 2013 have identified and classified bioactive phytochemical in medicinal plants and its biological functions to humans. That could be pertinent in analyzing constituents present on the leaves of Nephelium lappaceum and that of the commercialized tea. And this is the result of Abhishek et al.'s study. As we can see here, bioactive phytochemicals in medicinal plants are also attributed to different biological or significance to biological function. Vikramjit Kakati 2013 on the science of tea stipulated that Camellia sinensis is the plant from which all tea is made. Controlling the degree of oxidation during processing produces changes in chemical composition, color, taste, and scent. Furthermore, Kakati pointed out that tea includes a variety of compounds, but polyphenols, just like what the other researchers have stated, primarily, flavonoids are one of the most closely connected with flavor, fragrance, and of course, health benefits of the tea. And consequently, this study has worked on the following variables to yield the desired outcomes. As such, this study has aimed to investigate and seek the answers to fulfill the following questions. What are the active constituents present on the leaves of Nephelium lappaceum and Lipton yellow label tea and their corresponding RF values? Is there a significant difference between the active constituents of Nephelium lappaceum and that of Lipton yellow label tea? For methodology, as have priorly discussed, this study has employed a quasi-experimental research design in seeking to validate and explore the presence of active constituents in the leaves of Nephelium lappaceum, which is going to be compared to the active constituents present on the commercialized tea that will serve as the baseline for plants' potential as an herbal tea. And this is the research methodology or flowchart of the study. For the results and discussion, for section 1 on the active constituents present on the leaves of Nephelium lappaceum and Lipton yellow label tea and their corresponding RF values. For table 2, this indicates the active constituents present on Nephelium lappaceum's leaves and the Lipton yellow label tea. Double positive indicates the presence of a certain compound, while double negative hence indicates the absence of a certain active constituent. For essential oils, both Rambutan or Nephelium lappaceum's leaves and commercialized tea is positive with the presence of essential oils, while for phenols, it is only Nephelium lappaceum's has geared or has the presence of phenols according to vanillin sulfuric acid test. On the other hand, anthracenones for Lipton yellow label tea has been detected, and anthrones is only detected to Nephelium lappaceum's leaves, but not in the commercialized tea. But both test substances for phenols, tannins, and flavonoids have detected its presence for this particular test for blue spots. And of course, last but not the least, the presence of steroids. The presence of steroids has been detected on the commercialized Lipton yellow label tea, but not in Nephelium lappaceum's leaves. Next would be the relative flow rates or the values of bioactive constituents obtained from the presence of active constituents of each test substance. We have Rambutan and Commercialized Tea, and we have obtained the following computations based on the RF values. In connection to this, Vikramjit Kakati 2013 discussed that tea includes a variety of compounds, but polyphenols, primarily flavonoids, are one of the most closely connected with the flavor, fragrance, and health benefits. These compounds are produced by the plant to aid its defense against predators and stress, not for our benefit. As such, based on the gathered results on the test substances, both test substances, flavonoids has been detected for both the test substances on Nephelium, lappaceum, leaves, and Lipton yellow label tea. While Chandrasekhara and Shahidi stipulated that natural bioactive components found in herbal teas and beverages includes the following compounds, and of course one of which that is highly reiterated is polyphenolic compounds, particularly flavonoids. However, due to the unavailability and scarcity of test reagents and protocol for thin layer chromatography, some of these compounds were not tested and therefore were not able to be validated regarding its presence or absence on the test substances. So like what we have said, Peluso and Serafini also reiterated that this high level of bioactive compounds like polyphenols is assumed to be responsible for the health benefits attributed to its use. Like what I have said a while back, it can be said then that the presence of polyphenols, primarily flavonoids, can be a basis for the potentiality of a certain plant to be a herbal tea. Moreover, polyphenols, also known as flavonoids, are most likely one of the factors that contribute to these health benefits. But most generally, all teas consider or have the property of antioxidants to protect us from free radicals. For statistical treatment and analysis on the significant difference of the test substances, I have used dependent samples t-test to see if there is significant difference with regards to the detected presence of active constituents on the test substances. So I have obtained a value of 0.8204 or 824. Therefore, it can be implied that there is no significant difference between the test substances, at least at significant a level of 0.05. So there is no significant difference. This implies that the present active constituents to both test substances are completely similar in nature. In supplementation, Abhishek et al. 2013 have identified and classified bioactive phytochemicals, medicinal and its biological functions. So let's see let's use this as a basis in order for us to see whether there is significance of these bioactive components that have been detected to the leaves of Nephelium, lappaceum. So essential oils are considered to be antifungal and antioxidant. Phenols are antibacterial and antifungal. Anthrones, antioxidant. Phenols, tannins, and flavonoids are considered to be anti-cancer and antioxidant. So it is implied then that the presence of any kind of polyphenols to plant samples could be a guarantee of its usefulness and potentiality to be considered as an herbal tea. But of course, it is a multidimensional and multifaceted study. Therefore, this is just one of the considerations maybe that should be taken into consideration. For conclusion, The phytochemical screening and analysis on the leaves of Nephelium lappaceum and the commercialized tea have revealed that Nephelium lappaceum could be processed and considered as a tea based on the presence of beneficial constituents that make a tea a healthy drink. Statistically and descriptively comparing Nephelium lappaceum to the commercialized tea have paved the way to critically evaluate and assess the potential of Rambutan's leaves as herbal tea. With proper modifications, improvements, and rigorous research on the plant sample, herbal tea made out of rambutan's leaves may be considered and endorsed for production and consumption. And the following recommendations are indeed given to the following persons for chemists and tea experts in order to somehow uncertainties or to solve uncertainties of rambutan's leaves as an herbal tea will be comprehensively answered and systematically discussed prior to endorsement for tea production and consumption. For agriculturalists, wherein after a rigorous studies on the potentiality of rambutan's leaves that would increase and escalate the production of these trees, not just as an herbal tea, but any product that is of significance to human health. And lastly, for future researchers, since this study is bounded with limitations, scope, maybe the future researchers may consider to conduct different tests, just like anti-toxicity or toxicity tests and other related tests, including the assessment of different extraction methods that would generally contribute for the betterment and improvement of this study. So this is the guide for interpretation of TLC screening results that has been conducted for as a basis and guide for this research activity. And these are the actual photos of the plates that has been observed in order to obtain the gathered data. And these are the references of this study. And if there are any questions, clarifications, modifications, suggestions that you would like to ask, please feel free in order for us to have an interactive discussion. And in order for us, of course, to clear some concerns with regards to this sharing on my research activity. Thank you so much for actively listening.